Hi, welcome once again to our YouTube channel. Thanks for subscribing to our channel. I really appreciate it. Today we quickly want to take a look at the WAEC 2026 Biology Practical Specimen. I want to look at all the specimens that WAEC has released for this year's practical examination. And so we'll quickly have a rundown of all the specimens as WAEC has listed them out for us. So our specimen A, we have the groundnut pod. Groundnut pod is what we have for our specimen A. And for our specimen B, we have the crushed or roasted groundnut seed. Crushed or roasted groundnut seed, that's what we have our specimen B. Our specimen C is beaker with 200 ml of water. Our specimen D is beaker containing 20 ml of table salt solution. Our specimen E is beaker filled with sandy soil. Specimen F, we have it to be portion of alimentary canal of small mammal. Our specimen G is twig of a plant with mistletoe on it. Our specimen H is evaporated milk labeled in a beaker. We don't have specimen I in this particular list of specimen. We have our specimen J to be fresh fish. Specimen K is dry maize grain. Specimen L is tomato paste in petri dish. Our specimen M is fresh tomato fruit. Fresh tomato fruit. Our specimen N is rotten tomato fruit, which will also be put in petri dish. We don't have specimen O also, and then we have our specimen P to be thermometer. Specimen Q is meter rule, and specimen R is the quadrat. So those are the list of specimens that we have for this biology practical examination preparation class. So we quickly take a rundown at all of the specimens, starting with the first specimen, which is actually our specimen A, the groundnut. So what is the groundnut? Particularly looking at the groundnut pod here. So the groundnut pod, looking at the scientific classification, I put them up on the screen there by the right hand side of the screen in green ink. We can see the classification where we have the kingdom to be Plantae, we have the phylum to be Magnoliphyta, we have the class to be Magnoliphyta, the class there is Magnolipsida. We have the order to be Fabales, the family is Fabaceae, we have the genus to be Arachis, and then the species is Hypogae. And from the genus and the species, we have the scientific name of this particular crop, which is Arachis hypogae. So that is the scientific name or the botanical name. Now, what is this groundnut? Groundnut, widely called peanuts, is an annual legume or a pulse. Yes, it is an annual legume, or we can also call it a pulse. Now, this particular crop is found developing in so many environments, and it is found to be a root crop because it is found underneath the soil. Yes, it is a root crop, and then it grows up in pods. So we have the pods containing the groundnut seed. I'll show us a picture of that shortly. It is a dicotyledonous plant, so it has two seed leaves, and it has an hypogeo or surface germination. So its seed leaves are taken above the soil level, and that is why we refer to it as hypogeo germination. This particular crop is majorly planted for human consumption as well as used for animal forage because of its high protein value and fat content. It is eaten as snack with some other foods like gari and for preparing some native soups and delicacies. It is also used as raw material in the production of groundnut oil, groundnut cake, peanut butter, amongst other things. So these are some of the things that we use this particular specimen for. Now, showing us a picture of what the specimen is looking like, we have this particular picture there. We can see the brown pod having somewhat like the purple or the lilac color. So those seeds in purple or lilac color, they are the seeds of the bean, of the groundnut, while the brown is the pod of it. So you can see this is the flower. This is how the crop is when it is in the plant. So that is about our first specimen, and then we move to the next specimen, which is our specimen B. Still talking about groundnut seed. This time around we have a roasted crushed groundnut seed. Now this seed, after it has been harvested from the underground pods, they are shelled. Yes, we remove the shell because we do not eat them along with the shell, so they are actually shelled. We remove their shelled from the pods and then rinse thoroughly to get rid of the soil on it. So I'm just quickly going to talk about how the roasted crushed groundnut seed is being prepared. After you are done washing, you can actually add little salt to it. to taste if you like, and then you leave for a few minutes so as to allow the water in it to drain out. And then you allow, the reason why we're adding salt is to allow the salt to add little taste to it, and so we leave it for a few minutes to allow the salt to penetrate into the seed coat. So when we dry this particular crop, after drying, we put them into a hot pan, after which we roast for a couple of minutes, and then we can actually roast either using sand or we can roast using garri, and then it becomes edible. Now, processed or roasted groundnuts can be used to produce oil. Like I said earlier, we can actually extract oil from it, and it can also be eaten as snack. So that is about that. So we do not know how WAEC is going to structure their questions this year, but there is a possibility of having this particular specimen being used in a test or in an experiment. So I will put us through that shortly. So this is what the specimen that we're talking about. This is what it looks like. You can see this specimen here. So when it is being roasted with groundnuts, with garri, you can see how it looks. So that is how the finished product also look. So that is about the next specimen. And so we move over to the next specimen, which is our specimen C. Our specimen C, which is water, H2O. Water. So we have water in a beaker, and that is what our specimen C is. So what is this water that we're talking about? Yes, water is an essential substance that is colorless, it is odorless, and it is also tasteless. It is a neutral substance that has a pH of 7. That is, it is neither acidic nor is it alkaline in nature. It is made up of hydrogen and oxygen, and that is the essence of having the H2O written above. So it is made of hydrogen and oxygen. So what are the major uses of water? Number one, it is used domestically for multiple reasons. Example, for drinking, cooking, washing, as well as bathing. All of these are some of the reasons why we use water. And then we also use water to play key roles agriculturally, particularly when you need to water your crops. You use them to water your crops, you use them to clean your animals, to wash them, you use them in feeding your animals, as well as to dilute drugs or medications that is given to the farm animals. Also, we use water, which is very essential for normal functioning of biological organisms like plants and animals. So those are the things that just we need to know about water. So this particular specimen also could be used in an experiment that I will try to explain to us shortly. So we all know what water is, so I will just show us an example of water in a beaker. So we can see example of water in beaker. So that is what that is talking about. And then we move to the next specimen, which is our specimen D. Our specimen D is beaker containing 20 ml of table salt solution. Beaker containing 20 ml of table salt solution. So we are going to have some amount of salt, some amount of table salt added into water, and that is how we get the table salt solution. So now this table salt, what is the table salt? If you have to look at it, we have looked at water, so let us look at the components of the table salt. The table salt, also known as common salt, is what we refer to as sodium chloride. That is the scientific, or sorry, the composition of that particular substance. It is made of sodium and chlorine, so it is sodium chloride. It is a salt. Now, it is crystalline and it is highly soluble in water. And that is the essence why it can also dissolve in water that we'll be using for this particular study. So it is an essential mineral that is required in diet, very, particularly because we have majority of the salts produced these days to have iodine in them. So this same sample, that is the table salt that we're talking about, is dissolved, this particular one is dissolved in 20 ml distilled water to form table salt solution. Now the functions, it is very essential for nerve muscle functions. Yes, that's the function of the table salt. Now it is essential for nerve and muscle function. It is also used for seasoning, preservation of food samples. It is often iodized for health, and it can be used as raw material in the production of caustic soda as well as baking soda. All of these are some of the functions of the sodium chloride. Now, excess consumption of this particular substance is very bad because it can result to high blood sugar. So we have our salt here. When it is being dissolved in water, we can see what it looks like. So it gives us a colorless, clear solution, That is what the water is. On opposing to our specimen C, which is normal, just water. We said that it is colorless, odorless, and tasteless. But the specimen D now, it could be colorless, it could be odorless, but there will definitely be a taste as a result of the sodium chloride that has been dissolved in it. So you can see these are pictures of it. And so we move over to the next specimen, which is our specimen E. That's the sandy soil. What is our sandy soil? We know sandy soil is one of the three major types of soil that is found on the earth surface or on the earth crust. Others are the clay soil as well as the loamy soil. Now, sandy soil, if you look at the properties of it, is formed as the smallest fine particles of weathering rocks. When rocks are being weathered, when rocks are being broken down, that is how we get the sandy soil being formed. Sandy soil is characterized with high porosity and low capillarity. That is, it has little or no ability to take up water. That is what its capillarity is talking about, the ability to take up water. So it has low capillarity, and it is also very, very highly porous. That is, it drains water easily. Now, it is the poorest for agricultural practice. This particular crop is the poorest for agricultural practices because of its low nutritional value or fertility. It is very, very poor when it comes to agricultural use because it does not have the ability to retain water or nutrients as the plants will be needing them. This soil is denser than water, and so when it is mixed with water, it can easily settle to the base of the container in which it is placed. So we will look at how we can use this one also in an experiment. We do not know the dimension in which WAEC will be coming from, so we just try to cover all the bases as much as we can. So that is about that. So we can see an example of the picture of the sandy soil. You can see it in the picture there. So like I said, we could be having some experiments or some practicals, actual practicals put together by WAEC for these particular specimens. So we'll quickly look at what we can do with our specimen B, C, D, E in this particular experiment. Now, your specimen B contains fat because it is groundnut, it contains fat, it contains oil. So what if you were told to separate a mixture of oil and water? But first things first, let's start with the basics. Your fat there in the oil, you could be told to carry out a translucent test on your fat. So you carry out your fat test using a filter paper. You could be told to crush the groundnut which has been roasted already, and when you crush it, you put it in a filter paper. When you do that, you will discover that there is oil on the filter paper after some time. If you take the paper to the sun, or you look through the filter paper to the sun, you will discover that there is a part of the paper which is appearing to be transparent in form. So that is what we refer to as translucent test. That confirms that there is oil or there is fat in that particular substance. So that is test for fat on the groundnut, which is our specimen B. Now, if you have your specimen B dropped into specimen C, your specimen C is water. So if you have your specimen B dropped into your specimen C, which is having oil into water, you could be having something that looks like this on your screen. Now, that part can actually be separated out. If you have oil in water, you can actually separate it out. So how do you go about that? All you need to do is to get what we refer to as the separating funnel. If you pour this mixture into the separating funnel, you will be able to separate out, or you drain out your water. Since your water is denser than the oil, your water will be at the base of the container, and so you will just run off your water, leaving only the oil in the separating funnel. So that's how you can separate oil from water. Now, if you have your salt water now, and they ask you if you can separate the salt water, you can separate the salt from the water. Yes, that is absolutely very, very possible. All you will need to do is to carry out what we refer to as evaporation to dryness. If you evaporate your water, you boil the salt water, you eventually have your water evaporating out of it, leaving only your salt behind. So that's the way to separate salt from water. Now, you could also be told to separate sand from water. If you have your specimen E and you have to take it out of specimen C, let's assume you have a mixture of specimen C and specimen E. So in that kind of mixture, you find out that your sandy soil will go down into the water and be at the base of the container or the beaker. In that case, you can actually carry out what we refer to as decantation. Decantation is a process that you are seeing on the screen right now, where you actually tilt the beaker or the cup, allowing the water on it to run off, leaving the sand behind in the beaker. So that is decantation. Or better still, you can carry out what we refer to as separation using filter paper and funnel. You can actually do that as well to separate your sand from your water. Also, if you have a mixture of your oil, your sand, your water and your salt, is it possible to separate that as well? Yes, it is absolutely possible. All you need to do is to get your oil out first by performing decantation. You cannot use the separating funnel in this case here, because your sand will be at the base of it, so you cannot use separating funnel first. So you decant your oil out. After decanting the oil out, then you can now carry out what we refer to as filtration. You filter out to sieve out your sand. Remember that your salt is soluble in water, so your salt is already in solution with your water. So you can actually filter out your sand having your salt solution. Now when you get your salt solution away from your sand, remember that we have earlier on removed oil. Oil has been separated out. Now we have also concluded removing the sand, and then the next thing that you need to separate out of it will now be your salt. So you carry out evaporation to dryness. If you want your water, you can actually carry out evaporation to dryness in a closed circuit system, where your water will be collected in another container. So that is how you carry out this experiment. Perhaps you are told to discuss how all of this can be done. Now you have an idea of how to go about it. So we move over to the next specimen, which is our specimen F. So looking at our specimen F, we have it to be the portion of alimentary canal of a mammal. The portion of alimentary canal of a mammal. Actually, we have to prepare or make available a section of it that is from the esophagus to the stomach. But let me just run us through what that is talking about. What is this alimentary canal, if you may look at it? Alimentary canal is actually the digestive system of an animal. The digestive system of an animal is what we refer to as the alimentary canal. So this one is that of a mammal. So I'll show us example of that shortly. Now, it is the pathway through which food is taken into the body and broken down to derive nutrients, which is absorbed into the bloodstream. The nutrient that is required to carry on with daily activities is actually gotten from food that is being broken down. So the pathway that breaks down this food is what we refer to as the alimentary canal or the digestive system. Now, without wasting much of our time, I'll just quickly run us through some of the processes that take place in digestion, particularly in mammals. So foods are broken down, or they start their degradation right in the mouth, and that is where we have the first one there in the mouth. So if you look at the characteristics, I have there to be teeth. Teeth there is responsible for mastication, that is to chew the food that we put in our mouth. Some of the food that we eat cannot be swallowed just as we put them into our mouth, so there is a need for this food to be properly masticated or chewed upon, and so that is the function of the teeth. After which the tongue comes into play its own role of rolling the food into bolus form to make it easy to pass through the esophagus. And after this is done, there is going to be addition of saliva. The tongue mixes with the saliva in the mouth, and then there's an active component in the saliva which specifically acts on carbohydrates. That enzyme is called ptyalin. Ptyalin, P-T-Y-A-L-I-N. That enzyme acts only on carbohydrates. If you have any other food sample in your mouth when it is being chewed upon, be it fat and oil or protein, ptyalin will not act on those food. The function of ptyalin there is to convert the starch in the mouth to maltose. So that is the enzymatic reaction of ptyalin. It converts starch to maltose. So that is about what happens in the mouth, and then the food is taken into the esophagus. Now, before the food gets into the esophagus, there is a gateway between the mouth and the esophagus. We call that part the pharynx. I didn't want to bore us with too much notes, that's why I didn't put it in our write-up here. So the pharynx is actually a gateway that has what is called the epiglottis. That gateway permits substance to go either into the esophagus or into the trachea that leads into the lungs. The trachea leads into the lungs. So if food is to be taken into the esophagus, the epiglottis needs to cover the trachea, leaving the passage into the esophagus to be open, and that is when food coming from the mouth can easily go into the esophagus. So that being said, let us now look at what happens in the esophagus. The esophagus actually is a long muscular tube, and then food runs down that esophagus in a peristaltic manner. The food runs down through the esophagus in a peristaltic manner, and then that place is referred to as the passage of food into the stomach. So it is the passage where food passes through from the mouth into the stomach. Now, for the food getting into the stomach, it meets with an acidic content or acidic medium, which is actually made possible by the gastric juice. There is an active ingredient in the gastric juice, which is the HCl, hydrochloric acid. The hydrochloric acid makes the food to be sought to be acidic in nature, to prevent the food from rotting, and then to make it possible for the food to be able to continue its digestive or its digestion process. That is why the HCl in the gastric juice acts on it. Now, after that is done, the HCl also converts the food that is coming from the mouth, which has been acted upon by the ptyalin, for example carbohydrate or the starch, which has been acted upon by ptyalin, converting it to maltose, is now in an alkaline form. The food needs to be in an acidic medium so that every other digestive enzymes can act on it, and that is the second function of the HCl there. Apart from in preventing the food from rotting in the stomach, it also converts the food from the alkaline form to the acidic form, so that other enzymatic reaction or digestive reactions can take place on them. So those are the things that happen in the stomach. And then also we have the action of renin and pepsin in the stomach. Renin curdles milk, while pepsin acts on proteins, converting them to peptones. And then we move to the small intestine. The first entrance of the small intestine that we get to is called the duodenum. In the duodenum, that is where we have the bile emulsifying fat, and then we have the action of other digestive enzymes like the amylases that act on carbohydrates. We have the proteases that act, or the proteases or trypsins that act on proteins, and then we have the lipases that act on fats and oils. After these digestive enzymes have performed their actions, then the food has been properly broken down to their end products, which is carbohydrates broken down to glucose, fat and oils have been broken down to lipids, fatty acid and glycerol, and then proteins have been broken down to amino acids. All of these nutrients find their way into the second part of the small intestine, which is called the ileum. The ileum contains villi. They are finger-like structures in the small intestine that makes it possible for the nutrient to be sieved out of the small intestine into the bloodstream, so that the blood can actually take them to deficient organs of the body that require these nutrients or this energy. That particular site also absorbs water into the bloodstream. So that is what happens in the small intestine. And then we move over to the large intestine, where remnants of the small intestine content, all of the nutrients that has been sieved out, the waste that are left there, they are being pushed into the large intestine, into the colon. This particular part reabsorbs water, mixes with the waste products, and then it sends them into the rectum, where it is being stored as feces before it is being ingested out of the body. So that is about that on the digestive system or the alimentary canal of a mammal. So we can see the picture there. You can see what all of the enzymes that are being performed, all of the organs that carry out their functions. You can see where they are being placed and located. So this is a picture that shows that of man and the digestive system or the alimentary canal of a rabbit. So if you look at them, they are somewhat similar in nature. So that is about that. So we move to the next specimen now, which is talking about a twig of plant with mistletoe. A twig of plant with mistletoe. That is our specimen G. So what is a mistletoe? Mistletoe is a hemiparasite. That is, it is partial parasite. Why do we refer to it as a parasite or a partial parasite? It attaches itself to and grow on its host. It attaches itself to its host and it grows on its host, extracting water and nutrients from the host. That is why we refer to it as a parasite. It is benefiting from the host, but the host is not benefiting from it. Mistletoe contains chlorophyll. It is a chlorophyllos organism, hence it has the ability to actually generate its own food through photosynthesis. But this particular organism will not do that. It rather depends on the host completely for food or for nutrients. This particular organism gets water and nutrient from the host plant by penetrating into the tree trunks or the bark of this host. It penetrates into the bark with specialized root-like structures called the ostomium. So it uses its ostomium to actually pierce through into the bark of the tree, and then it starts to derive nutrients and water from the host. It is often found on trees like oak tree. We have it on apple, neem, guava trees, etc. So those are examples of what the mistletoe on its host plant looks like. So you can see here are some pictures of what this particular plant looks like. So those are just pictures of it. And then we move to the next specimen, which is our specimen H, talking about evaporated milk. Evaporated milk is gotten from the milk actually, but when it becomes coagulated, when it's actually is being processed, that is when it becomes evaporated milk. So it is another dairy product formed and gotten from farm animals like cattle, pigs, sheep, goats. All of those bovine animals, they are the ones that produce this particular milk that we're talking about. Animals produce milk in their udder and is extracted from milk for mature females. It is extracted from mature females. It is only the mature females that have the ability to produce milk. It is actually extracted from their udder. We actually use a particular specialized equipment or machine to actually extract this milk from them, from the udder of the female animals. So I'll show us a picture of that shortly. Some of these animals are reared specifically for the purpose of milk production. Yes, we have so many animals that are reared solely for that particular purpose. The milk is rich in protein, in calcium, etc. We have iron in them also. It is processed into cheese. It can be processed into cheese, into yogurt. Milk that has not been pasteurized could contain disease-causing pathogens, and that is one of the essence why people actually need to pasteurize their milk to prevent them from causing them any harm. So we have these pictures here which will help us understand what we are talking about. So this is what the milk looks like. You can see when it is evaporated. So that is about the specimen H. And so we'll move over to the next specimen, which is our specimen J. Specimen J is fresh fish. Fresh fish from the freshwaters or freshwater habitat. Now, for this particular tutorial, I used a particular specimen, a particular fresh fish as my specimen, and the fish that I used is the tilapia fish. So we have other types of fresh fishes as well, like we have the mackerel, we have the salmon, we have the croaker, we have the catfish. All of these are fresh fishes because they are gotten from freshwater habitat. But I just decided to streamline this discussion to tilapia fish, and then I will use that to actually explain what I want to talk about. So if you look towards the right here, to the right hand corner of the screen, I put up the classification or the scientific classification of this organism. The kingdom there is Animalia. We have the phylum there to be Chordata because it is a chordate, it has backbone. The class there we have the class there to be Actinopterygium, and then the order is Cyclophamis. We have the family to be Cichlidae. We have the genus to be Tilapia, and the species Spamani. And so that is how we get the scientific name of tilapia fish to be Tilapia spamani. So like I told us, we have other types like I listed here: mackerel, salmon, croaker, catfish, etc. So what are fresh fishes? Freshwater fish, they inhabit hollow waters. That is, they are found in rivers. More suitably found surviving in warm waters than in cold waters. Obviously, some of these fishes, you find them in warm waters than in cold waters. One of the healthiest source of protein is this fresh fish that we are talking about. It actually has high amount of proteins. They have high amount of proteins in them. They can live up to 10 years and can weigh up to about 10 pounds. That is how much they can do. They can become big. Now, all of this, majority of them, with the exclusion of a few of them, they have scales. I said the exclusion of few of them, like the catfishes do not have scales. The body of the catfishes are very, very slimy. So, but the tilapia fish that I'm using as an example here, they have scales, they have streamlined bodies, they possess fins. We have different types of fins. We have the lateral fin, we have the dorsal, the ventral, the pectoral fin. All of these fins aid their movement in their habitat. Now, these organisms make use of the gill system for respiration. They actually use of the gill system for respiration. In the underneath their operculum, you have their gills there, which is used for respiration. Now, these organisms are oviparous organisms. They are oviparous in nature, that is, they lay eggs, and they have high resistance to diseases. If you look at the commercial advantage of this particular specimen, they grow fast, and that makes it very, very possible for you to actually make heavy returns from them. So they grow very, very fast. And then we have them feeding on algae and zooplankton. So it is not, they do not always wait for the food that is being given to them, particularly if you are rearing them. You can actually get them algae and zooplanktons. If they are living in a somewhat a natural habitat, they can actually get that to feed on. Also, they have high adaptability, so they can actually survive almost in any environment that you get them to. And then we also call them omnivores and then cannibalistic in nature. Yes, because they can feed on their kind, they feed on flesh also, so they can also feed on their kind, particularly if they feel threatened or starved. So that is about the fresh fish. So like I just said, I used tilapia fish as my case study here. If we're talking about tilapia catfish, where I said that the tilapia fish possess scales, for catfishes I will say that they do not have scales, but majority of the other things, the other characteristics are similar for both catfish and tilapia fish. So you can see some of these examples here. You can see these examples here. All of these are fishes, they are freshwater fishes. So that is about that. And so we move to the next specimen, which is our specimen K. And specimen K is dry maize grain. Z maize is what we call it, the dry maize or maize grain. We call it scientifically to be Z maize. So if you look closely down here in blue ink, down the screen by the right, I put up the classification of this particular crop there. Classification there, the kingdom is Plantae, the phylum is Trachyphyta, the class is Liliopsida. We have the order to be Poales, family is Poaceae, the genus is Z, and the species is maize. Hence the name called Z maize. What is maize? Maize is a staple crop. It is a cereal. It is a herbaceous crop. It is an ephemeral. It's an ephemeral because it completes its life cycle in 3 to 4 months. This particular crop is a hypogeo monocotyledonous plant. It is a hypogeo monocotyledonous plant. So it has its leaves, its seed leaf remaining in the soil. Its seed leaf remains in the soil. It is rich in fiber. It has high amount of fiber. So that is one of the reasons why it is being consumed. It is the world's most produced crop cultivated for humans and animal foods, as well as industrial use. The pH requirement of this particular crop, particularly if you want to cultivate it, is about 5.5 to 6.5 pH. So it is slightly acidic, weak acid. So we have different types of this particular specimen. We have the yellow maize, we have the white maize, we have the popcorn, we have the sweet corn. All of these are examples of what the maize look like. So that is about our specimen case. So here are some pictures that can help you understand what we are talking about. You can see these are pictures to help us understand what we are talking about. So that is about that. And so we move to the next specimen, talking about specimen L. Our specimen L is tomato paste. Yes, tomato paste. So what is our tomato? Scientifically, we call it Solanum lycopersicum. Solanum leucopassicum, that is the scientific name. So classifying it, we have the kingdom to be Plantae, the phylum is Angiosperma, the class is Eudictota, we have the class there to be Eudicots, we have the order to be Solanes, family is Solanum, Solanaceae rather. The genus, we have it to be Solanum, and then the species is Leucopassicum. That is why we have this scientific name to be Solanum leucopassicum. Now, this particular crop is gotten from ripe tomato. This particular specimen now is gotten from ripe tomato fruit, which is processed to become edible. And how do we go about that? When we harvest the crop, the tomato paste, the tomato, we wash properly and then we mill or we blend it to form a paste. Now, after that has been done, we actually steam it or we parboil it to make that particular crop to be edible. So we steam it, and after steaming, then we can actually go about eating the crop, the specimen. So we steam the specimen, and when it is properly steamed, it gives us what we have there to be the tomato paste. Now, some people might add little oil or little additive to it, preservative to it, just to increase its shelf life. So this is used in meal preparation, and then particularly for making stews, sauce, etc. So that is what this particular specimen is about. So these are pictures of what the paste look like. You can see these are pictures. That is what this paste look like. And so that takes us to the next specimen, which is our specimen M. Specimen M is fresh tomato fruit now. So we have talked about the tomato fruit, which is we've talked about the paste of the tomato fruit. So let us look at the tomato fruit itself now. This particular one, I'll show us a picture of it. You'll see that it is very, very firm and fresh. It is a dicotyledonous plant with hairy stem, and we have the pinnate. It has hairy stem and pinnate. It is often cultivated as annual crop. We cultivate this particular crop as annual crop. Now, it is classified thus agriculturally as follows. We classify it agriculturally as follows. One, we have the fruits. The fruits to true fruits. We have simple fruits. We have succulents. We have berry. So those are the classification of this particular crop. It is classified as fruits, true fruits, simple fruits, succulent, and then berry. That is the classification of this tomato fruit. Tomato is low in calories. It has low amount of calories, and then it is very rich in vitamin C, vitamin K, potassium, antioxidants, beta carotene, amino acids. All of these are nutrients that can be gotten from tomato. It also has high amount of water in it. It has very, very high amount of water, and this content of it makes it or gives you the ability to aid digestion and help maintain healthy cholesterol levels. So that is about the tomato fruit, the firm tomato fruit. You can see that particular specimen. It has fresh outer coating. You can see it. That is how the specimen looks. And then when this particular specimen is not properly stored or preserved, we can then have what our specimen N is, which is a rotten tomato fruit. Rotten tomato fruit. So tomato starts to rot due to a physiological disorder, which soon degenerates due to microbial infections caused by fungi and bacteria. When microbial infections like fungi, like bacteria, when they start to attack the crop, they can leave it to become messy. Some of these causes of rot include, we have the blossom end rot, we have the fungal pathogens, we have the bacterial soft rot, etc. The symptoms, the commonest symptoms you get to see is that it becomes water soaked, leathery brown or black spots that I've seen on the crop and at the base of it. And then there's going to be a breakdown of the cell wall, rupturing of the cell wall. All of these are so similar. They are symptoms that are experienced or that are seen on the substance or on the specimen when it starts to rot. All of these leaves the fruit messy. Yes, when it becomes infected by either fungi or bacteria or whatever, It leaves the crop to become, or the fruit to become, messy and smelly, making consumption of such impossible. So that is about that specimen, which is our specimen N. So you can see how that look here. You can see these are pictures of how that particular specimen look. So that is about that. And then we move to the next specimen, which is our specimen P. Our specimen P. Talking about our specimen P, it is a thermometer that is used to measure the degree of hotness or coldness of a body or of a substance. That is our specimen P. We do not have specimen O, so we have specimen P to be thermometer, and the thermometer is used, like I just said, to measure the degree of hotness or coldness of a body. We use it to check the temperature of a substance. Now, it measures the boiling and freezing points during experiments. You can actually use it to measure degree of freezing or boiling during experiment. We use it to measure temperature ranging between -10 degrees Celsius and as high as 110 degrees Celsius. So that way we can get 0 to 100 degrees Celsius without any H. So we have different types of these thermometers. We have the digital thermometers. Digital thermometers, they have screen where the unit, or sorry, whatever measurement that is gotten is being displayed on. And then we have the ear thermometer, we have the ear thermometer, we have the strip thermometer, and then we have the infrared thermometer. All of these are examples of thermometers, and so you are seeing them here right now. That is what the thermometer is looking like. All of these are examples of thermometers. You can see them. So that being said, you could be asked to draw the thermometer. You can see this is how the thermometer looks. If you are asked to draw, that is how the thermometer look. So we can actually do that. Draw if we are told to draw. You can see, and label properly. So we move to the next specimen now, which is our specimen Q. Our specimen Q is the meter rule. What is a meter rule? The meter rule is a handy tool typically used to measure distance, length, or sizes. We use it to measure distance, length, or sizes. And there are different types of meter rules, and that they measure in millimeters, centimeters, and meters. Some meter rules are longer but flexible, and they consist of a case. Some of them have thumb lock, we have blade, we have tape, we have hook, and sometimes belt clip. Some are made from wood, some are made of plastics, fabrics, metal strips, etc. A tape usually have imperial reading, metric readings, and then both on same tape. So we have some of the tapes having both on the same tape. So those are some of the characteristics of the meter rule that you need to know. So we use them, like I said, to measure in meters, be it millimeters, centimeters, or meters actually. So that's about the meter rule. So you can see here, these are some examples of what the meter rule look like. So we then move to the next specimen, which is our specimen R, which happens to be the last specimen that we have for today's experiment, or today's tutorial, specimen R, and it is a quadrat. So what is a quadrat? A quadrat is a population study or sampling instrument that is used to estimate the number of organisms that is found in a particular environment. We use it to actually check the number of organisms that is found in an environment, and that informs how we study organisms in that environment. So we use them, we toss them at random to get some frequencies of some organisms, and then we work based on the results that has been gotten to actually estimate or to have a check on what is going to happen in that population size. Now, quadrats are square or rectangular objects made from different materials like wood, we have metals, plastics, PVC for example. All of these are substances that can be used or that have been used in the past to produce what the quadrat is. Now, this object is tossed at random on a selected plot. We must have selected a plot, pegged it, marked it. So you now pick a quadrat and you toss at random, not to the same location, you toss at random to different locations on that same plot. Now, a couple of times, you can toss that a couple of times, let's say about 10 times or 20, depending on what readings you're working on how you want to get it. Now, this process can be used to know the population size, it can be used to know the population frequency, the percentage cover, amongst so many others. So those are the functions of the Quadrat. It is an ecological instrument. So you could be asked now, based on this question now, if you look at this now, we have our specimen P to be thermometer, specimen Q is meter rule, and specimen R is Quadrat. All of these are ecological instruments, so I'm suspecting that we could be having more questions coming on ecological instruments and then their functions. So please let's make ourselves acquainted with that. So that being said, that brings us to the end of this tutorial session. I implore you to try as much as possible to go through what we have discussed again. Get yourself familiar with what you have discussed. Help yourself to know and understand all of this better. Perhaps you have any question that you need clarity or you need help with, you cannot fail to put them down in our comment section. Put them down in our comment section. I will try as much as possible to get back to you as soon as possible on them. For those that have come with us all along, we really appreciate you. Thank you very much once again. If you are the first time out to our channel, please click the subscription button and click on the notification button as well, so you get notified when we put up new content on the channel. Please send this video to send the link to your friends. Let them also benefit from what you are getting, because I know this is going to come handy to you as you do your examinations. All I wish you is success in the exam. Thank you very much, and bye for now.